Hello everyone, so welcome to Glycolysis Part 2. So today will be a really short one, summing up some of our stuff on glycolysis, but also introducing the stuff we'll be looking at more detail. So I'll recap from the last lecture. So we defined glucose oxidation or cellular respiration as 4 pathways. So we had our glycolysis, pyruvate oxidation, citric acid cycle, and electron transport chain. Again, this is synonymous with cellular respiration. We talked about glycolysis and how glucose oxidation is essentially a method of getting glucose into a cell. We also talked about methods of getting glucose into a cell. We had our facilitated transport using the GLUTs. Specifically, we talked about GLUT4. We also had intramuscular glucose uptake and cell-mediated glucose uptake. We also talked about co-transport with sodium using the Symport mechanism. We also talked the goal and overview of glycolysis, looking at the 10 steps and the 3 different stages: initiating, priming, and splitting. We looked at our Gibbs standard for energy and looked at our reversible and irreversible reactions, as well as naming the enzymes. So again, here's a recap. We have our priming, splitting, and oxidizing reducing stage, and all those steps we took at from 1 to 10. So again, it's important to know what's really happening. So remember that when we go through glycolysis, it's really important to follow the carbons. So remember that glucose has 6 carbons and pyruvate only has 3. This means that 2 pyruvates are formed for each glycolysis. So remember, it's splitting here and it's actually happening twice. So net yield energy of energy in glycolysis and cellular respiration. So first, we need 2 ATP required per glucose. So we need 1 in here and 1 in here. So remember, we talked about glycogen. If glycogen is used instead of glucose, it enters here. So we use 1 less glucose. Again, so 4 ATP are generated per glucose in glycolysis, and there's a net gain of 2, because we use 2 and gain 2. But like I said, there's a 3 ATP net gain from glucose from glycogen, since it enters at glucose 6-phosphate, which is right here. So again, remember glycogen can also enter glycolysis. Glycogen enters at step 2, meaning it requires 1 less ATP. This means it's more efficient energy source than glucose and involves a net 3 ATP gain. And then you can see here it moves in the same system, going through citric acid cycle and etc. So here's a quick summary of glycolysis. So again, go back through the notes in our summary of the last lecture of each of the 10 steps. So again, what's inputted into glycolysis is 1 glucose molecule. We need 2 ATP, and we need 2 NAD+, as well as 4 ATP. This is to go to ATP. Our outputs or products include 2 pyruvates, which are those 3 carbons, 2 ADPs, because we use 2 ATPs that are now ADP, so those go to there, 2 NADH, which become oxidized, 2 NAD+, that become oxidized to NADH, and then we have ATP, which goes to 4 ATP, and as well as 2 water molecules. So it's usually important to know each step, substrate, product, reaction type, enzyme name, and the rate limiting steps and the summary above. So it's really important to know these inputs and outputs, as well as the limiting steps, so which ones are reversible, irreversible. So one of the key ones we always see is phosphofructose. And then a lot of the enzymes and then the products of each step. So the regulation of glycolysis. So there are certain rate-limiting enzymes in glycolysis. These include hexokinase, also known as HK, which is step 1, and phosphofructose, or PFK1, in step 3. So here we have fructofructokinase and our hexokinase. So step 1 and step 3. So the energy status of the cell controls the activity of phosphofructokinase 1. Again, so when you think of energy status, you can think of exercise as certain exercise decreasing our energy stores and stuff like that. So again, the impact of exercise and exercise training can affect the activity of PFK1. So another summary. So where does glycolysis take place? It takes place in the cell cytoplasm. Why? Why does glycolysis take place? So essentially, it splits glucose to yield ADP, to produce reducing equivalent NADH, to produce ATP via oxidative phosphorylation, and produce pyruvate substrate for phase 2 of glucose oxidation, which goes into our citric acid cycle. Glycolysis can use glucose or glycogen, and remember, glycogen enters at step 2. When glucose occurs all the time and is always happening, but the rate of glucose depends on the demand for ATP. So an example would be rest versus exercise. So obviously during exercise, we have an increased demand for energy. So we need more ATP. There's an increased need for ATP, so glycolysis rate increases. Again, there's only 10 enzymatic reactions, so glucose is a relatively fast metabolic pathway. So again, this is bringing it back to kinesiology. So here essentially are 3 energy pathways. So we have our aerobic and our anaerobic and also our glycolysis. So essentially, this figure, so here we have exercise duration on the side and percent energy, ATP essentially, or speed. So we can see at the timing, so it's immediate exercise, so I start to run. We have our glycolysis working immediately, but even before that, we have ATP creatine phosphate. So these are usually located within the muscle. This is our immediate energy. So this is the second we move, this is what's being used. And this is mainly seen or highly studied in high intensity activities, so sprinting, jumping, stuff like that. So you can see it only lasts maybe up to 30 seconds. And even at that time, it's kind of already run out. So it's that kind of 15-second mark. Then we have our glycolysis kick in, which is our short-term energy. So again, we can see that lasts for around 2 minutes. Then it starts to fade. But then we have aerobic, where this is our slow long-term, and this uses is our long-term energy. Again, one thing that people need to remember is that they don't occur one at a time. They're all working at the same time. It's just one is more predominant over the other at different times. So as we mentioned, phosphofructokinase as that one of those rate-limiting enzymes is very important and dependent on energy status. So again, phosphofructokinase traps glycolysis at step 3, making it irreversible. Exercise can increase the speed of phosphofructokinase 1, improving the Km. So the Michaelis constant. So changes in the ADP to ATP ratio. So again, if we have higher ADP, it means in a lower energy state, and then we have higher ATP in our cells, it means we're in a low energy, a high energy state in the sense that we're not using it. So phosphofructokinase 1 is one of the most important regulating enzymes in glycolysis. It has 4 subunits and is a quantitative protein, and PFK is allosteric. PFK is inhibited by high ATP at low pH associated with exercise when there's increase in hydrogen ions. And this inhibition is reversed with AMP. So PFK regulates glycolysis through allosteric inhibition. So the cell can increase or decrease the rate of glycolysis, or the Km, in response to the cell's energy requirements. So again, it can detect whether there's a high ATP or high ATP, and then determine what's its energy status, and then up or down regulate glycolysis if needed. So again, this is the end of our short lecture. So this was part 2. So next we're looking at part 3, and we'll be looking at the fate of pyruvate in the next lecture. See you next time.